So, good morning, participants. Now we are moving toward the second session of 1 week e-FDP day 1. We are very fortunate to have a speaker, Dr. Yogeshwar Bacchav, founder and director of Adex Pharma, Mumbai. Dr. Bacchav have academic credentials like he is a doctorate in pharmaceutics and postgraduate in pharmaceutical chemistry from Indian Institute of Chemical Technology, Mumbai. He did his B.Pharm from North Maharashtra Mumbai University and he did his law from University of Mumbai. He also did a global MBA from University of Western Australia and a certification course of EMDP B3 from IIM Kozhikode. Currently, he is a director and pharmaceutical development consultant at AI Curie's Anti-Infective Cures AG, Germany, and he is a founder of Adex Pharma R&D consultancy. His work includes 18 years of experience in pharmaceutical R&D and drug development. His expertise across the entire new drug development life cycle. He is a leading in uh, chemistry, manufacturing, and control development of 30 plus molecules from early to late clinical stages. He managed around 22 plus successful investigational clinical phases. His area of expertise and interest includes pharmaceutical development and CMC, new drug development and life cycle management, formulation and analytical development, regulatory affairs and market access, project management and business development, strategic and commercial decision making. His research and academic contribution leads to around 18 peer-reviewed research publications, 7 international patents, and he contributed to the books and conference in presentations. He is an active member of Control Release Society USA and American Society of Pharmaceutical Scientists. His Google Scholar profile indicates his citations are above 1300, his H index is around 12, and I index is 16. So we have a very good speaker for the second session, and he is going to deliver a talk on formulation challenges to deliver water insoluble drugs for parenteral administration. So on behalf of all of us, now I request sir to please start your session.
Hello, can you all hear me?
Yes. Yes,
Yeah,
Yes, sir,
can I
you're
start?
audible. You can share your screen.
yes.
Yeah, sure.
Oh,
You
okay,
can
I
share
can
yours.
start. Yeah, sure. So, thank you for a kind introduction. It is my pleasure to be here and to deliver a talk. The topic I'm going to talk today is formulation of parental formulations for the complex molecules. Can you all see my camera, or I'm not
No,
starting, right?
no, sir. No, sir.
Okay, let me start it.
Yes. Now you are
Now,
visible. Yeah.
okay. So I hope my slides are visible. You can see the details, right?
Yes, sir,
Yes.
your slides are visible.
Okay,
You may start with your session.
okay, sir. Thanks, ma'am. So basically, we start with the topic: why do we need a parental formulations in the course of drug development or discovery? So, as introduced by ma'am before, my experience over 2 decades comes from the drug discovery, basically the drug development, starting from the scratch until the preclinical development, different phases of clinics like phase 1, phase 2, and phase 3, followed by the commercial launch. So often in the early phases, like preclinical phases, we have to establish the PK profile in order to determine the absolute bioavailability. We also need to do proof of concept studies where the IV route is preferred. There is also need for tox studies if the proposed clinical route is IV route. For example, the drugs given in the ICU settings. Of course, in ICU, nobody would like to have the tablets or capsules, right? The drugs need to be given by the parenteral administration. So I also listed in the here like clinical phases like using hospital antibiotics, transplant patients, oncology injectables, and that's where the need for the parenteral formulation is must. Having said that, oral route is always preferred route when it comes to the drug discovery because it's easy to develop. It's the cost effective for the patients. They can take at home. They don't have to go to the hospitals. But if at all parental is needed, then it's needed. There is no bypass, and that's where then the complexity arises. So when we say complexity, what are the complexity reasons? So poor solubility is something most important factor we all need to consider because in the drug development phase, the low solubility of the drug is really something obstacle for the formulation scientist. And if you see the graph, the majority of the drugs in the pipeline they are poorly soluble, and this really leads to quite significant challenge to develop not only oral but also the parental formulations to develop as well as scale up, and especially when you have to deliver the drug in quite high amount. The doses are quite high for hospital antibiotics. Imagine you have oncology injectable where dose is just 10 milligram, maybe etoposide or paclitaxel. Then I mean milligram amount of compound is not something, let's say, difficult to solubilize. You don't need quite high amount of excipients. But the same situation you can consider for the antibiotics like antibacterials, antivirals, where the doses are quite high. Typically antibiotics, the dose is quite high, 4 to 5 gram per day. And if the drug is not soluble and you have to use some excipients, then life becomes quite difficult. Then you are forced to use some solubilizing excipients like surfactants, co-solvents, cyclodextrins, and then due to the low amount of excipient concentration allowed by FDIG limits, then it's really difficult to come up with the suitable formulation, and that's where the complexity arises. So also, I mean, there's one option, you want to keep the API in the solubilized state, but sometimes also particle size reduction could be considered, maybe nanosuspension, but that is again not so easy. It is quite complex process, complex operation, so this is to be carefully evaluated. And here you have some distribution that the whatever marketed drugs you see around, let's say 45, I mean almost 70% of the drug they present the problem of the poor solubility, right? And for the drug development, you go to the drug development alone basis class 2 and class 4, which is poor solubility matrix. 90% of the drugs are in that domain. So having said that, one must address the problem of poor solubility in order to develop the same and efficacious formulation. Also, apart from that, what are the other advantages of developing new formulation strategies? So, of course, you can reduce the cost, and this is true for the very expensive APIs like paclitaxel. Maybe then you have improved formulation. This also gives you the possibility of IP protection. You can also extend the patent life. Dose can be reduced, and also the dose-related toxicity can be avoided. And having done all this, of course, this leads to the patient compliance. So these are the benefits you can consider. What are the critical quality attributes when one has to develop the formulation at early stage? When I say early stage, it's really before clinic, when you are developing the preclinical formulation. So selection of the excipients is something quite important, because whatever excipients you will select at this stage, this will have impact on your cost, this will have impact on your process, this will have impact on let's say the any potential drug excipient interactions. So this has to be very carefully considered. And having said that, any excipient you use, this must be approved by FDA. This the concentration you are going to use, this must be within the IG limits. So as said before, drug must be solubilized form when you go for IV route. In some cases, you may go for the insoluble state of the drug if you are going to use the nanomilling or nanoparticle approach. That is an exception. So typical excipients we use are the solubilizers, buffering agents, antioxidants, pH modifiers, and to optimize the osmolality. The acceptable pH for the parenteral formulation, that it must be around 7.4. But if you see the many marketed products, they do have the pH between 3 to 9. So that really depends. But if you go about pH 9, it may lead to tissue necrosis. If it is acidic pH, then it can also lead to pain or phlebitis. So you have to be carefully optimized. Then what is the injection route, whether it's IV or intramuscular or subcutaneous, what is the total volume you are going to deliver. So there are many factors, so these are factors you need to consider to decide the final pH. Typical osmolality you need to have between 280 to 290 milliosmoles per kg, because this is the osmolality of the blood. But it's not also uncommon to have the formulations slightly hypertonic. Hypotonic should be avoided, but again, this has to be carefully considered what is the volume you are going to inject. So there are some exceptions that you can go for the hypertonic formulations. Sterilization is something quite important, because any formulation you are going to use for the injectable route, it has to be sterile. So whatever process you are going to develop, it must be suitable for sterile filtration, aseptic processing. Normally, the authorities will ask you to go for the terminal sterilization. So when I say the terminal sterilization, the formation must be subjected to the autoclave, 121 degrees Celsius, 15 psi for 15 minutes. Majority of the formulations are not stable to this temperature conditions. If the drug is prone to the hydrolysis, if it is a peptide or protein molecule, it may undergo degradation. So in that case, you need to write necessary justifications to the authorities that terminal sterilization is not possible, and you can go for aseptic processing, aseptic filtration. But imagine if your formulation is highly viscous, or maybe your drug is bound to the filter, maybe then you are not able to aseptically filter your formulation, and that is a drawback. And this is something you have to carefully consider quite upfront, so that you do not experience any hiccups at the later stage. Whatever API and excipient you are going to use, you just have to pardon me for 1 minute. Sorry. And whatever excipients or API you're going to use, they must come with the low bioburden. And immune stability is something you need to consider whenever you have like stock formulation. Imagine you have the vial containing 20 milligram per ml as a stock, maybe in 2080 or polysorbate or propylene glycol mix, but it needs to be diluted in saline solution prior to the infusion, and that is called in-use stability. So you have to basically dilute the stock formulation using that vehicle, and there you need to demonstrate that upon dilution, the pH is not impacted, the osmolality is not impacted, the drug is not precipitating out. No, this is the data authorities would like to see. So far so good. Can you guys still hear me? I mean, I have a feeling I'm just talking to myself, so you just need to confirm if all good so far. If anybody can say if so far all good. Hello? Can someone raise hand or say something so that I know I'm not talking to myself? Hello? Hello? Hello?
Hello.
Can you still hear me? I just needed some confirmation that you guys can
Yes.
still hear me. This is ongoing. Otherwise, I just feel I'm talking to myself, and then I just need to
No,
know if
no, no.
all.
You were audible, and your screen also was good visible, sir.
Perfect. Perfect. Thank you very much.
Yeah,
Thanks for this.
now it's perfect, sir. You can continue.
Thank you. Thank you.
Okay, sir.
So for any formulation to be developed, then you must set the target product profile. This is something authorities would like to see. So here I just tried to put an example for antifungal drug. So there are two factors to consider. I mean, what is the desired product attribute and what is must. So when I say desired, it is like wish, this is like I would like to see. But must is something that there is no option around that, so you must try to fix it. So for example, a dose is something, 50 to 100 milligram, but then it must be less than 300 milligram. Dose frequency desired is once per day, but it should not be more than 3 times a day. Duration of treatment, like 1 week is desired, but it must, less than 2 weeks. So you have different consideration for pH, for tonicity, for use of excipient. So this is the target product profile you must set before the development. This must be approved by your project management, senior management, as well as the clinician toxicologist, and that helps formulator to develop the product. So typically the packaging type you're going to use, maybe plastic bottles, the glass bottles, what is the storage temperature, what is the shelf life you're expecting. So ideally I would like to see 36 months, but if not, okay, you can also go for 24 months. Whether we have freedom to operate for use of any excipients, whether there is any IP infringement, cost of goods should also be considered, whether we have to involve any capex at the third party. So these are the typical considerations for target product profile. Now the interesting topic, what are the strategies we should consider to formulate the poorly soluble drugs? So I already tried to put some of the advanced technologies normally we use to develop the formulations for the poorly soluble drugs, like pH modification, use of co-solvents and surfactants, then developing lipid-based formulations like microemulsions, intralipid emulsion, liposomes, mixed micelles. You can also use cyclodextrins for complexation, nanosuspension, something important, and then finally the pH adjustment. So let's start with the pH modification. So basically, for any ionizable compound, if it shows the pH dependent solubility, so you can always take the advantage of the pH modification. So good example, you can take ciprofloxacin. So this molecule shows pH dependent solubility, and it is highly soluble under acidic conditions. So they tried to develop the IV formulation using lactic acid as a pH modifier, and this helped to improve the solubility. Another option you can always consider at very early stage to prepare salts. So imagine if your drug is really basic, then you can go for the salt formation using acidic counter ion. Other way around, if your drug is acidic, then you can use the basic counter ions to come with the salts, and the resulting salts are normally highly soluble. So here is another case study. So they tried to prepare the soluble salt of aspirin. Aspirin is like age-old molecule. You will see a lot of indications of this molecule. The researchers are still working with this molecule. So lastly, they wanted to use aspirin for the treatment of migraine as an option to sematriptan. But again, they needed quite high dose, and aspirin was not soluble at that particular dose. So basically, they just prepared the DL-lysine salt. So DL-lysine is basically amino acid. So and the resulting salt was highly soluble. So and then that's how they tried to use this salt, at quite high concentration. And this was effective compared to sumatriptan. But precaution to be taken, or one has to be careful using this approach, that there may be in vivo precipitation, because what happens, this drug, once it is dissolved in water or saline, the pH will be acidic because you are using the acidic counter ion. But the moment it is injected, it is mixed with the plasma of blood, and the blood and plasma pH is 7.4. So due to the rapid pH shift, there may be precipitation. So this is something to be avoided, and for that you need a lot of in vitro data to show that there will be no risk of in vivo compatibility. Then we talk about co-solvency or use of surfactants. The best example you can take of paclitaxel. The conventional formulation of paclitaxel into the market was using the combination of cremophore-EL and alcohol. But cremophore-EL was prone to induce some hypersensitivity reactions. And then prior to the injection of this paclitaxel, they needed some antihistaminic treatment. And there this company, they came with this abraxane in US, albumin macrospace, which contains paclitaxel nanoparticles, in fact, or nanosuspension. This was a replacement to the Taxol formulation in order to eliminate the cremophore-EL. So problem with this approach, you use the co-solvents quite in high concentration, and this may lead to the adverse events. And the mechanism it works, basically it reduces the dielectric constant, and due to that, there's enhanced solubilization of the poorly soluble or non-polar drug molecules. Often you can also use the co-surfactants in combination with surfactants or pH modifiers. That really helps to increase the overall solubility. Another example you can take of docetaxel, which is another anti-cancer drug. And this formulation was a taxotere, which containing around 54% of polysorbate 80, around 40% of dehydrated alcohol. But due to very high concentration of these solvents or surfactants, there was also incidence of hypersensitivity reaction. And that's where the players like Sandoz, Hospira, Apotex came using this 5,5B2 regulatory pathway, and they tried to use the solvent like PREG 300 to replace twin 80, and these resulting formations were quite safe and stable to the innovator taxotere. But again here, caution to be taken that due to high co-solvent or surfactant concentrations, there could be poor tolerability issues. Once this co-solvent formulations are injected, there could be incidence of the precipitation, and this needs to be avoided. And here, just for information, I tried to include the different marketed formulations for the parental route, which use normally the solvents, and you can also see the typical routes of administration, which drugs they have used, and at what concentrations these excipients are used. Then moving to the cyclodextrins, how do they work? So basically, you have the water insoluble compound. This is a typical aspirin-like structure you see here. So this is water insoluble. So basically what you do, you use the excipients like cyclodextrin, which are water soluble, but they do have the cavity, which is a hydrophobic. So basically the hydrophobic part of the molecule fits within this cavity, and the hydrophilic part remains out, and so the resulting complex is water soluble. That's how it works. This is quite easy process. You can also work with quite small API quantities. And you see in the market, there are several cyclodextrin based formulations available for parental use. The typical cyclodextrins which are approved for parental route are hydroxypropyl beta cyclodextrin, hydroxypropyl gamma cyclodextrin, Captisol. Use of Captisol is more and more these days because of the adverse event with other cyclodextrins. I tried to put some case studies here using the Captisol. So you had a molecule like Vesti patent, which was a selective neurokinin receptor antagonist. This was mainly used for the treatment of post-operative nausea and vomiting. And the formulation from innovator was a mannitol-based formulation, but it had quite hemolytic activity in the preclinical studies. And this was not due to the excipients, but this was the inherent property of the drug, that it was leading to hemolysis. So what they did basically to take care of this hemolytic potential, they prepared the drug cyclodextrin complex of vesipatin with Captisol. So the part of the molecule which was responsible for hemolytic part was sitting inside the cavity, and the resulting complex was no more hemolytic. And then thus they tried to address this problem hemolysis using this complex formulation, which was quite interesting. This is another case study for Captisol, which was a investigational formulation. Again, the compound they used amidaron, so which was developed for IV route, again to take care with the cardiac, for the indication of cardiac arrhythmias. But there was an incidence of systemic hypotension because of the presence of the solvents, co-solvents like polysorbate 80 and benzyl alcohol. So basically, the innovator formulation was coming with polysorbate 80 and benzyl alcohol. So to minimize hypotension, they diluted this IV formulation with the distilled water prior to the administration, and it was slowly infused. So what happened basically? This was still not able to take care of the hypotension issues. So they came up with this PM101, which was a novel formulation, and kept its solo use. So in this case, they did not use any polysorbate 80 or benzyl alcohol, and when they tried to use this complex, there was no any hemodynamic effect, which was mainly coming from polysorbate 80 or benzyl alcohol. And this is something they confirm in the beagle dogs. So the outcome of this study shows this new formulation PM101 and its vehicle, it produced no hemodynamic effect. On the other side, there was quite rapid and significant hemodynamic activity due to the presence of polysorbate and benzyl alcohol alone. And here you see the commercial parental formulations where captisol used as a solubilizers, just for your information. Of course, the list is increasing, just to be sure. Then let's talk about the lipid-based formulations. So lipids are also quite important platform for the formulation development. So there are several lipid-based formulations you can explore for the delivery of poorly soluble drugs for IV route. But for that, your drug has to be have quite high locality, or at least the partition coefficient must be high so that the drug shows some affinity for lipids. So, for example, if your drug is quite hydrophilic or low locality, it may not be soluble in the lipids. So, this is something to be carefully checked. So, that may be the reason you may not see any lipid-based formulation for peptides or proteins, because they are highly hydrophilic or water soluble, and they will not be normally soluble into the lipids. Another important consideration you have to do that your drug must be stable in contact with the lipids. That is quite important, because majority of the lipids they have the residual oxidizing impurities, and then due to the presence of these oxidizing impurities, the API might undergo degradation, and this is something to be avoided. And then, of course, lipid is not something soluble in contact with water. So in order to emulsify lipid in water, you will need the surfactants. So the quantity of surfactants you'll be using has to be carefully considered. Typically, the lipids will not induce any osmolality or contribute to any osmolality. So you have to also make sure that osmolality building agents to be added to make sure that your formation not is not hypotonic. And typical examples of lipid phase formation used for parenteral administrations are microemulsions, liposomes, intralipid emulsions, and mixed micelles. So let's go one by one. So microemulsions are typically oil, surfactant, and water combination, and these are like isotropic mixtures. So to the naked eye, it looks like a clear vehicle. Yeah? And this is quite widely used technology because it is simple to manufacture. And this is quite important delivery system which can be used to deliver the insoluble drugs. So typically they can these systems contain 10 to 20% oil lipid droplets, then surfactants like 20 to 30% remaining is water, and as said before, osmotic agent to be added. In case if it is a multiple dose container, then preservative needs to be added. And this slide shows the different marketed microemulsion based formulations for the parenteral route. Typical case study you can consider the propofol injection. So this is quite classical example of parenteral microemulsion formulation. For this drug, first they used cream for EL, but then later on ethanol was used to replace cream for EL. But again, we are discussing the presence of co-solvents. Surfactants always has some issue with respect to any hypersensitivity reactions or other adverse events. So it's ideal to remove any surfactants, co-solvents if you can, come with some advanced formulation. And that's where this propofol microemulsion was quite helpful, where they use soybean oil. Mixed micelle is also quite important platform here. So basically, you try to mix two surfactants like poloxam or lecithin. So you just mix this with API, all three ingredients, or not ingredients, but three parts like API and two excipients. They need to be dissolved in the organic solvent. You get a thin film. It needs to be carefully hydrated with the buffer so that you get the mixed micelles made up of lecithin, poloxamer, and drug is sitting into the center. This is a case study quite interesting I did during my time in CIS. So what we did basically, you get it ready to use mixed micelles from the market. At that time, at least we used to get. Now it's no more into the market. So I dissolved my drug into ethanol, and this ethanolic solution was added to the ready to use mixed micelles. In the beginning, you see there's a clear precipitation of the drug due to the rapid mix, but when you gently shake it over the period of time, finally you get a quite decent mixed micelles which was ready for the parenteral administration. I was able to get drug loading of 2 to 5 milligram per ml. And this really helped us to develop formulation for the preclinical studies, because normally we get around 40 to 50 compounds, and then you go on developing IV formulation for every molecule. This is not advisable in terms of time and budget. So availability of ready to use kit was very useful that time. This is another case study for curcumin, so where they tried to develop the mixed masses for curcumin, and data here is quite important that for free compared to free curcumin means drug was not encapsulated; it was simply dissolved in some solvents, and then they developed the mixed masses using lecithin, sodium docosate, or pluronics, and the data showed that compared to the free curcumin, the mixed micelle formation showed the better exposure, and this is mainly due to the stabilizing effect of the mixed micelles onto the molecule. Liposomes is another important platform. This is quite significantly used for small molecules as well as peptides, proteins, antibodies. So if you see the structure of liposomes, so these are basically the vesicles, like, and you have the lipid core where drug can sit, and you have the central hydrophilic core, the aqueous core where molecule can also be solubilized. So the beauty of liposomes, you can use it for both hydrophilic as well as hydrophobic molecules. But if it is a lipid molecule, so basically it will be sitting within the lipidic bilayer. So, and the process you are using to develop liposomes is very important. That also drives where your molecule is sitting. What is the drug loading is something important. So you will see a lot of liposomal based IV formulations. They are being used in clinical trials already onto the market. And typically, it is quite expensive technology, so you'll not be using it for simpler indications. It is mainly used for the chronic indications like cancer or maybe complex diseases like liver malfunctioning. The important case study in this case is doxorubicin. You will see the liposomal formulations of doxorubicin in the market. This drug basically works by inhibiting the DNA and RNA synthesis, but this drug also has a strong affinity for the heart lipid called cardiolipin, and due to this, there's a heart failure. Basically, the cardiotoxicity is quite severe. So what they did, basically they tried to develop the liposomal formulation, and this led to the discovery of two products, myosin and doxil. And here is data I'm trying to put. Compared to free drug, the doxil and myosin was able to increase the bioavailability quite significantly. The half-life was also quite significantly higher for the liposomal product, and the incidence of adverse event was quite low compared to the free drug. So having said that, when drug was formulated as liposomal formulation, the incidence of adverse event was much lower compared to the free drug. And these are the typical formulations, liposomal formulations you have onto the market, just for information. In terms of timekeeping, I'll talk for 5 minutes more, and then we can take question answer for last 10 minutes, just to be sure. Then the last lipidic platform will discuss about intralipid emulsions. So these intralipid emulsions are also quite important. They have been used for some time for the clinical use, and this mainly serves as an energy source for the hospitalized patients because these patients are not able to take the drug by the oral route, and that's where these lipids serve as a source of fatty acids and vitamins. There's quite high interest recently to deliver this intralipid emulsions by IV route because then lipid soluble drugs are dissolved in lipids, they are micro emulsified, not micro emulsified, but emulsified, and this good compatibility of the lipids with the tissues, so there is no excipient toxicity. So typical components of intralipid emulsions are the lipids, like MCTs or LCTs. Emulsifiers are like typical lecithins or PEG modified phosphatidylethylamine, polyoxamers, and of course you have water. So what are the advantages? So there is no pain or irritation at the site of injection. There is no toxicity or reduced toxicity. You can induce high stability in solubility for the drug. Targeted drug build is possible, but you also have disadvantages basically. So you can't really go for quite high drug loading because if your drug is not soluble in lipids, then it's problematic. Uh, there could be instability during the storage if your drug is going under degradation in contact with the lipids or surfactants, and there's no possibility to use quite a wide range of SEPs emulsifiers, so choice is quite narrow there. And this is typically processed. So you dissolve your drug in oil. This is mixed with the aqueous phase with surfactants. You go for high shear mixer. Then you get a coarse emulsion, and this coarse emulsion is then subjected to high pressure homogenization, and you get a fine emulsion. And then you subject this formulation to the filtration. So the cutoff is 5 micron. So anything below 5 micron is acceptable for the parenteral administration. And typical process parameters you'll consider rate of mixing, temperature, the cooling, pressure, number of cycles, and duration of sterilization. So at the end, you'll subject these vials to the terminal sterilization, as we discussed in the beginning. And these are the typical market drug which contains injectable emulsion, just that so the drugs, type of drugs they have used, the products, and the final composition. So that might help you to consider which are the oils and surfactants are permissible to develop the intraliquid emulsions. And then the last approach, I'll talk about nanosuspension. So we discussed in the beginning for paclitaxel, they came up with this Abraxane, which is a paclitaxel based drug delivery system. So basically, it is a nanoparticle based product which was approved by FDA in 2006. So this is a novel formulation, and it comes as a lyophilized formulation. And drug loading is 10%, 90% is albumin. The final particle size is 130 nanometer. So the maximum tolerated dose observed using this formulation was much higher compared to this cremophore based formulation. So having said that, this formulation was a very good alternative to the conventional formulation. In terms of process, they used the bottom-up technologies. So basically, drug and excipients were dissolved in organic solvents, and there was a controlled precipitation. So basically, drug was entrapped within this albumin matrix. And of course, if you want to go for alternative methods, you can go for a high pressure homogenization or media milling. Atocone is another case study if you want to consider. So this is typical inhibitor of T gondi as a organism, but it shows quite poor absorption after oral route. So so they came with the IV formulation, as by oral route it was not possible to get the bioavailability, and they developed the nano emulsion formulation, nanosuspension formulation using the stabilizers like polysorbate, poloxamase. So when they tried the concentration higher than 1 microgram per ml nanosuspension, so there was no cytotoxicity, in fact. So basically they were able to rule out any cytotoxicity, but at the same time they got good efficacy. So this nanosuspension route provides you quite good opportunities, like it can go for patent life cycle management. You can deliver quite high amount of dose because you are not using any excipients to solubilize the drug. The excipients you are using, they are quite safe, but the problems or challenges are, this is a difficult technology to scale up. The control on particle size is very important. The stability on storage is something you have to carefully consider, and sterilization is something important. So this is the last slide I will discuss, and then maybe we can go to the question answer. So one of the important approach you have to consider for parenteral administration is the Adex compatibility studies. So when the drug is developed as a stock solution into the vials or maybe plastic containers, but it has to be diluted again prior to the administration into the infusion bag. So authorities will ask you to show the data when the drug is diluted into the given vehicle, maybe dextrose solution or saline solution or water for injection, whether the drug remains stable, whether there is any absorption of the drug onto the container, onto the closures, onto the infusion line when it is stored in syringes. So that data to be generated for, depending on the storage, like maybe 6 to 12 hours at room temperature or under refrigeration conditions, that there is no drop in assay on purity, there is no absorption of the drug onto the containers, there is no shift in the pH, there is no presence of any visible, subvisible particulates. This is the data you have to generate and show to the authorities. That is called Adex compatibility data, and this is quite mandatory. Another important aspect is extractable leachables, where you have to demonstrate that your formulation is not extracting any extractable leachables from the bags you are using, from the rubber closures or the infusion line. So this is also key data they want to check at phase 3 or for the commercial formulation. Having said that, in the interest of time, I'll stop here and I'm happy to take any questions you might have. How I can stop sharing my screen.
Yeah, you can. Hello, sir, are you there?
Me? Hello?
Yes, you are audible, sir.
Sorry, I was kicked out of room. So I'm open for any questions you might have.
Yeah, yeah. Sir, question is visible on screen. Can you see? Should I read?
No, no, I can read. Yeah, I mean,
Yes.
see, it's quite.
Huh.
Your question already has an answer, So you say
Yeah.
why chemical ethanol was selected as a vehicle instead of simple co-solvent system. So if I'm a formulator, if I will start something with simple, but if simple co-solvent system is not able to deliver the desired formulation, naturally I'm forced to use the chemical and ethanol. So you will always go stepwise. Start with something simple. If something simple is not working, then you go for more complex issues, but the complex issues should also have the answers then.
Yes, sir. I think there are no more questions. So, Dr. Yogeshwar Bacchav have detail explain about strategies for poor water soluble drugs. He included many examples and case studies for pH modification, co-solvency, lipid-based methods, micelles, microemulsions, liposomes, nanosuspensions, etc. So, on behalf of principal and the organizing team of e-FDP, I am Mrs. Smita Shirke. Uh, feel honored to put on record a big thanks to Dr. Yogeshwar Bacchav for sharing his valuable knowledge from his busy schedule and accepting our invitation. Sir, kindly accept our certificate of appreciation.
Thank you, ma'am. Thank you for this one. Yeah.
Yes, sir. Thank you very much, sir, and have a good day.
Thank you, ma'am. Thank you, everyone. Bye.
Bye. Third session. Participants are informed that third session will start at 12:15.